Hello there. So, B1 paper, hiya. Let's do it. So by this time, you should have had a go at these questions. If you haven't, but it's not the end of the world, but it'd be nice for you to have something down, then you can mark it or correct it accordingly. So, standard paper, 60 minutes, 60 marks, 6 questions, getting harder as you go down the paper. So let's begin. Disease. The photograph shows a housefly. Housefly is an animal vector of disease. Dysentery, we know this. This is one of the examples the examiner wants you to know. The disease dysentery is caused by a bacteria. Describe how a housefly can cause a person to become infected with dysentery. So it's a vector, so it's going from one place to another. Now, dysentery is usually from feces, animal waste, contaminated animal waste, and then it lands on your food and transfers it. So we've got to put that into words. So housefly lands on animal waste that is contaminated. Dysentery is transferred onto food, which is then eaten. Horrible thought, but how many times has a fly landed on your food? Lots. And this probably just, well, it's obviously it's a video of dysentery or not, but there is that chance. So, let's turn over. 3 marks. So we're going to explain how a chemical defence mechanism can reduce the chance of dysentery. So what chemical defences do we know? You should know 2. Lysosome and HCl. And HCl is going to be the one we're going to use because we're not going to need us crying out bacteria to stop it, are we? So, hydrochloric acid, you can put HCl or after that in the stomach, kills bacteria. Or if you want to put dysentery there, that's fine. So, that's 3 marks, 1 mark, 1 mark, 1 mark, killing. Okay, so sometimes you have to be a bit weird like the last question, going around the houses, but we needed to. This one, bang, bang, thank you, ma'am. Let's move on. Explain how what's this word? I don't know. Oh no, this one there. So don't worry about it. It's going to be words, especially when you do classification. You might not come across. Don't stress about it. Okay, mosquito can spread the disease malaria. So, same thing. The mosquito is a vector, just like the housefly was, and carries. This is tricky though, this bit, because you need to know that it carries Plasmodium, which is the disease part, and which is transferred to the blood when the mosquito bites. So really, that's not a nice question as a whole, question number 1 really. But if you go with a question like that, it's a bit tricky. You can't lose your head over it. Okay, you would have picked up marks, that's a fact. You just got to move on. Okay, come back to it, have a little think about it later. Right, this question is about classification. How do that tells us there? So, have that in mind. It's all about animal kingdoms. We've got 2 tick the boxes here. So the first one, which animal kingdom do these organisms belong to? So you've got to know what dictates these different classifications. Well, you know the animals and plant cells have a cell wall, and you know that fungi is multicellular, so you're left with Protista. You've just got to know your characteristics. Got your nucleus, etc. Right, in which structure of the chromosomes are these organisms found? just bread and butter stuff, that is, people. You just need to know that. Cilia are hairs, chloroplast is photosynthesis, flagellum are the propeller parts of a cell. Just one reason why Euglena and company have been placed into the kingdom Plantae. So what have they got in common with the plants? Go back, chloroplast, wham, bam, thank you ma'am. And then also, you've got to think what else makes them, what else makes a plant a plant. But you're only suggesting one. So I just go chloroplasts are present. Oh, cheeky. Chloroplasts are present. It's unusual because it's both heterotrophic and autotrophic. Explain how this helps Euglena to survive. So say, this question is asking you, do you know what that means? Do you know what that means? Yeah, of course I do, yeah. So, autotrophs. So, how does that help us out? Autotrophs have the ability to make their own food by photosynthesis using the sun. Right, then I'll just go for heterotrophs. Heterotrophs eat other food. Therefore, can live in different environments. So if it's got the ability to do both, then that's fine, isn't it? If it's not sunny, I'm going to get some food. If I live in a warm environment, fine, I can just autotrophically make my own food. Scientists discovered new species in boiling acid mud in Costa Rica. Explain how this discovery can be validated by the scientific community. So you make a finding, do you know what you have to do then in order for everybody to say, actually, yeah, he's right. So, then I'm just going to do this as a flow chart. You publish in a journal, then that is peer reviewed. And we've got 2 marks already then. And then you could also attend conferences. It's a cheeky few marks there, and it's the chance of it turning on your paper very slim. But if they are, you just need to know. If you make a finding, how do you get that out there into the wider scientific community? Those are the reasons, or they are the ways. Plant growth substances. Oop, there. Right then. Photograph A shows the root of a plant when it's positioned horizontally. The root was kept in the dark, left in this position. Photograph B was taken 3 days later. Complete the sentence by putting a cross in the box next to your answer. The process that resulted in the type of root growth is sown. Right then. It's going down towards gravity. If it's going towards gravity, it's positive. So it's going to be positive gravitropism. It was out of these two. Because these two mean light. It's not about light, it's about gravity. So it's you recognising that positive means towards gravity. 3 marks now. Explain how this change in root growth has occurred. So it's step by step. We've done this in your book. You know that first of all, auxine lies on the underside of the root. Auxine in roots inhibits growth causing the root to grow towards gravity or downwards. But you've done the diagrams. You've got to learn it, people. Learn your stuff. You get the answers right then. Dead simple. So, suggest question. Suggest question. What's the point in having that? What is the point in the plant doing that? So it can be anything really. It's 2 marks. So you just want to say that it enables the root to what can we use? I'm trying to get across it. It's always going to keep it in one place. So anchor enables to anchor the plant in the soil. That's where soil is, where gravity is. Also, it enables it to reach water and minerals. Wham bam, thank you, ma'am. Right, turn over. Not turn over, shift it aside. You've got a practical here. You get given practicals and you've got to try and figure out what the hell's going on. So let's see if we can figure out what the hell's going on here. Rebecca and Andrew set up each experiment to investigate the effect of light on plant shoots. They also set up control. Rebecca placed a black cap over the tip of the plant. Andrew removed the tip from his plant shoot, placed a thin block of jelly on top of the plant shoot, and then replaced the tip. So you've got covered block of jelly, nothing. First thing, why do we set up a control? So, compare. Compare results to normal conditions. Because without that, you don't know what the plant's going to respond like if you do nothing to it. So you need that as a control, so you can compare it. Well, if we didn't do anything to it, then this is what happened, and then so on, so forth. Right, so explain the results of Rebecca's experiment and Andrew's experiment. So 3 marks. So we're going to need a bit more than 1 point on each. So, what happened and why did it happen? Rebecca's experiment, it didn't grow towards the light. That's what happened. What does that tell us? That tells us the thing responsible to make it bend towards the light must be in the tip. Which is similar to Andrew's experiment. So let's just have a look at, let's do Andrew's first, Rebecca's first, and we'll crack on Andrew's in a second. Because there's a point we need to make. Rebecca's shoot did not curve, 1 mark for making an observation. This shows auxine must be in the tip of the shoot. So it didn't move, so the thing we're making it responsible for move, it's got to be there. Then Andrew's experiment. Shows the jelly had no effect on growth, meaning the auxine could move to the shaded side. So that's just saying it had no effect. And telling them a lot of that was out of shot, I do apologise. So Andrew's experiment shows the jelly had no effect on growth, meaning the auxine could move to the shaded side. So I'm just saying what auxine does in normal circumstances. Okay? Question 4 already, people. The skin. Looks like temperature regulation to me, does it you? Brilliant, because we can do all these all day long because they're easy peasy. Right, X, blood capillary, dermis, nerve ending, sebaceous gland. It is a sebaceous gland. You see how at the beginning of these questions you're getting these 1 marker recalls. Okay? Elimination, dermis means skin, nerve ending, no, it's not going to be that. You've got to live in the diagram basically. You either know it or you don't, do you? Describe the role of the sweat gland in thermoregulation. So what does it do? Sweat gland releases water, which evaporates and cools the skin down. 3 marks there. 1, 2, 3. For a 2 marker. You see again, succinct. Get to it, man. Stop waffling. Right, turn over. So let's have a look. See. Explain why the muscles attached to the hair follicle is important when the person starts to feel cold. So, what happens in the cold to hair, and how is the muscle involved? So, hair stands up on end when it's cold. The muscle contracts. It works. The muscle contracts. Hair stands up on end, which traps a layer of air next to the skin, acting as an insulator. Let's put that down, eh? The erector muscle contracts, causing the hair to stand on end. End doesn't begin with A. This traps a layer of air which acts as an insulator. And that's more than ample for 2 marks. Again, 3 marks again. Thermoregulation is quite easy, I think. Thermoregulation is one way which constants maintain. What was in England to me? Homeostasis. Come on, ask me one I don't know. Dear me. Explain why the temperature of the human body needs to remain at 37 degrees. Now this is quite tricky. And it's really looking from B2, and it's a good job you've been taught B2, because I probably would have neglected to say this and would actually deliver B1. Because you know enzymes function best at their optimum temperature, which is 37 degrees. And that is absolutely fine. Done. Enzymes, optimum temperature, you've got your marks there. If you want to say it above this, they begin to denature, fine, but you're picking up marks and you've already picked them up. Explain why the reptiles lie in the sunlight for long periods of time. What do we know about reptiles? Well, we know they are poikilotherms. I should have put in the proper sentence. So reptiles are poikilotherms, which means they cannot regulate their own temperature and rely on the sun. Done. Turn over. Pollution. We're on question 5, and it's the first time we've got a bit of data. Unusual, but get used to using data. I'm going to need a calculator, I think. Right, the diagram shows the river. Location of first back to the arrows indicate the direction. You've got the river coming down here. You've got a factory. You've got site A, site B. What have we got in our sample? Looking at nitrate concentrations, A and B. A, mean. Calculate the mean found at site B. Oh, dear me. 49 plus 64 plus 58 equals 171. How many 3's in 171? I'd imagine it's going to go, so hopefully it does. None in that. 17's 5 in that. Carry 2 it goes. 57. Easy peasy. Of course, use a calculator, please. So it was that sum divided by 3. Oh, done that one. Nearly there, people. Nearly there. Stick with me. Stick with me. Got to do 2 sets markers yet. Right. Scientists observed algae in some dead fossils at the site B. They are not present at A. Give an explanation for these observations. That means there must be more nitrates at B, I'd imagine. Of course there are. So there's more nitrates at B. So let's write that down first. More nitrates leaked into the river at site B. This causes eutrophication where algae rapidly grow, blocking sunlight to underwater plants, causing them to die. Dead plants are broken down by microorganisms. These in turn respire, removing oxygen from the river. And then everything else dies then, but we've got so many marks here. So you've got 1, 2, 3, 4, 5, 6. 6 before there. Okay? Eutrophication. It's drilled into you. Just got it. Make sure you don't miss the opportunity to express it. Right, describe how level of water pollution and air pollution can be assessed using living organisms. Oh, what a dream of a 6 marker. Water pollution, air pollution. We've got to look for paragraphs on 6 marker. We've got 2, water pollution, air pollution. But then we've got another 2, haven't we? Because you can have clean water, dirty water, clean air, dirty air. So 4 paragraphs. Paragraph 1 I'm going to talk about clean indicators, then dirty indicators, and then clean for air and clean for dirty. So we've got 4 beautiful paragraphs coming right at you. Okay. So, indicator species, let's do a nice little introduction. Indicator species allow us to determine levels of pollution. Nice little opener there for you. So, if water is polluted, you will find organisms such as bloodworms or sludgeworms as they live in deoxygenated water. However, if there is a presence of freshwater shrimp, spelled wrong, or stonefly, then this indicates the water is not polluted. Done. Now, I'm not going to do this last bit because you get the gist of it. But if you carry on in that vein, but for air pollution, you've got to talk about two things: black spot fungus only grow in unpolluted areas. Because sulfur dioxide kills them. And then you've got also lichens, which again, different lichens, different species can survive different amounts. So by the species of lichen, you can tell the levels of pollution. That's 6 marks done. We're on a dream here, I tell you. Because this paper isn't that hard. Let's carry on then. Last question. Power through. Power at home. Run 26 minutes. We'll have it done in 30, half the time. It was about that, eh? State the level of classification of both parts. The binomial name. First name, genus. Second name, species. Genus. Species. Some rats have a mutation that allows them to eat rat poison, warfarin, and survive. Suggest how the warfarin... It's okay.